Are you trying to study physiology and feeling lost? In Unraveling Human Physiology, I will try to make physiology easier for you to understand. I choose body acid-based regulation because I know it's a very difficult subject to understand. As it is complex, I divide the explanation in several videos. Look at all of them and then you will be able to integrate and understand as a whole how the body managed to regulate body acid-based status. In this part, we are going to discuss why is pH so important. Turn your TV or look at some videos and I'm sure someone will be talking about pH. This shampoo has a balanced pH. This water has neutral pH. What's the pH of these fruits? An acid pH will ruin your crops. It's important to control the pH of your aquarium. Why is pH so important? So let's start by defining what is pH. pH equals minus the logarithm of hydrogen ion concentration. So when we are talking about pH, we are talking about hydrogen ion concentration. And why are hydrogen ions so important? Is it because they are big? No, they have a tiny mass, just one atom with no neutrons, only protons. That's why we usually call them protons. Are protons important because there are many in the body? No. Let's do the math. Normal extracellular pH is about 7.4. If pH is minus the logarithm of proton concentration would be 10 to minus pH. 10 to minus 7.4 is 3.8 times 10 to minus 8 equivalent per liter. So that is 0.0000038 equivalent per liter. Try to read this in an analysis and understand if your patient has a normal extracellular protons or not. That is very difficult. So it's a lot easier to understand if you transform this number to pH. That will vary between 1 to 14. How is this concentration if we compare to other ions in the body? Normal extracellular proton concentration is 0.00038 milliequivalent per liter. Sodium, which is the most abundant cation in the blood, has a normal extracellular concentration of about 140 milliequivalent per liter. So if we compare those two, you would see that sodium is about 3,500 times more abundant than protons. If the concentration of protons is 1 gram of salt, the concentration of sodium would be 3.5 kilos of salt. So let's put it in a more graphical way. Let's go to the kitchen. Where do you find a gram? A gram of salt would be about a teaspoon of salt. 3.5 kilos of salt is very difficult to put together in the kitchen, so let's choose another thing. 1 kilo of apples is about 4 apples. So 3.5 kilos would be 14 apples. So graphically, the concentration would be for protons a teaspoon of salt and for sodium 14 apples. Then why is it so important? It's about its reactivity. In water, hydrogen ion is not really free. It's a tiny mass with a positive charge, resulting in a molecule of high density of positive charges that will attract any electronegative molecule. In water, oxygen is very electronegative because it has two uncoupled electrons that will attract protons. This results in a new molecule with a coordinate bond with the proton called hydronium. So, in water, hydrogen ion plus water equals hydronium. So remember this key point. Hydrogen ions are very important because they are very reactive. If you combine two molecules of water, you will obtain hydronium and oxydril. So In a way, it's like water is acting like a base and hydrogen acid conjugate acid. Interestingly, hydrogen doesn't stay in the same molecule of water but change partners many times per second, giving the impression that hydrogen travels. That gives the solution the property of conductivity. Here's another key point. Remember that the concentration of hydrogen and oxidril are related. If pH lowers, that is, the concentration of hydrogen rises, then the concentration of oxidril will lower. On the other hand, if pH rises, that is, the concentration of hydrogen lowers, then the concentration of oxidril will rise. If we talk about pH, we have to mention acid and bases. So let's start with the acids. What happens if we add acid to water? If you add acid to water, you will obtain hydrogen and the conjugate base of the acid. That will, of course, raise hydrogen concentration and will lower pH. That's why we call acids proton donors. Acid can be weak or strong depending on the magnitude of acid molecules that will dissociate in water. An example of weak acid is carbonic acid. Carbonic acid will dissociate in water to form hydrogen and bicarbonate. How do we know it's weak? We calculate the acid dissociation constant of this reaction. The acid dissociation constant of this reaction is the concentration of hydrogen times the concentration of bicarbonate divided by the concentration of carbonic acid. In this case, it's very small, 4.4 times 10 to minus 7. Again, as in pH, we will transform the acid dissociation constant to pKa, applying minus the logarithm of the acid dissociation constant. For example, in this case, if acid dissociation constant is 4.4 times 10 to minus 7, pKa will be 6.4. We know that an acid is weak if the pKa is between minus 2 to 12. An example of a strong acid is hydrochloric acid. Hydrochloric acid will dissociate in water to form hydrogen and chloride. The acid dissociation constant of this reaction is the concentration of hydrogen times the concentration of chloride divided the concentration of hydrochloric acid. As hydrochloric acid is a strong acid, almost all the molecules of hydrochloric acid in water will be dissociated. Then the concentration of hydrochloric acid will be very small. If we divide by a small number, then the acid dissociation constant will be big. In this case, it's 1.3 times 10 to 6. We will apply minus the logarithm of acid dissociation constant to have pKa. pKa of this reaction is minus 6.1. You see a very small number. We said that if the pKa of the acid dissociation constant is less than minus 2, the acid is strong. And what about the bases? If you add water to a base, you will obtain the conjugate acid of the base and oxidril. That will raise the concentration of oxidril. But remember, if the concentration of oxidril rises, the concentration of hydrogen will drop. If the concentration of hydrogen drops, pH will rise. And that's why we call the bases proton acceptors. Base can be also weak or strong depending on the magnitude of the molecules that will be dissociated in water. We have weak bases where only a fraction of the bases will be dissociated in water, and we have strong bases where almost all the base will be dissociated in water. As with the acids, we can analyze if the base is strong or weak, calculating the dissociation constant. But in the case of the bases, we call that the base dissociation constant. We also calculate minus the logarithm of the base dissociation constant, pKb. If pKb is between -2 and 12, we say that the base is weak. If pKb is less than -2, we say that the base is strong. So remember this key point. If the pKa of an acid is less than -2, we say that the acid is strong. If the pKa of an acid is between -2 and 12, we said that the acid is weak. Now on, for the matter of simplicity, instead of talking about hydronium, I will talk about protons or hydrogen ions. But bear in mind that there are not free hydrogen ions in water. Many organic compounds have acid or base groups, so if body pH changes, their ionic groups will change. If pH changes, many body processes can be affected. For example, proteins are formed by amino acids. Amino acids are compounds that have an amino terminal group, which is a base, a carboxy terminal group, which is an acid, and a side chain R that could be either neutral, acid, or basic. When an amino acid is free, you will have in an equilibrium the carboxy terminal group in the acidic protonate form, or its conjugate base without protons. And in the amino basic group, you will have an equilibrium with the form which is not protonate and the acid form which has more protons. During peptide synthesis, the amino terminal group of an amino acid reacts with the carboxy terminal group of another amino acid to form the peptide union. In this process, water is lost and both extremes are joined. They don't behave anymore like acid or base. But you still have an acid in the one extreme of the peptide and a base in the other extreme. To know the overall charge of a peptide or a protein, we have to consider the charge of the R side change. R side change can be either neutral, acidic, or basic. So the overall charge of a peptide or a protein is the combination of the charges of all the amino acids that form this peptide or protein. Proteins are not just a sequence of amino acids, but they tend to fold into the space to form three-dimensional structures. These structures are very important because they determine the function of the protein and also the way one protein interacts with another protein. An important factor to determine what is the three-dimensional structure of the protein is the R change of the amino acid that conform the protein, because they can be positive charges or with negative charges. If pH changes, these charges could change, and this would lead to a change in the structure of the protein. The 3D structures won't be the same, and that can result in a loss of function of the protein or a loss of interaction protein with another protein. There are many ways in which pH can change a protein. It can change the structure, it can change the interaction. If it is an enzyme, it can change its function. If it is a channel or a transporter, it can change its activity. Sometimes the change is because of a change in the protein structure, as I explained you before, but sometimes the change is because just of a change of charges in a tiny little place. Every protein has an optimal pH where it can function, and this pH depends on the protein. For example, the optimal pH of a lysosomal proteins, which is a place which is acid, will be acid, and the optimal pH of a protein in the blood, which normally is 7.4, would be 7.4. Proteins are not the only compounds that can be affected by pH. For example, in lipids, we have triglycerides. Triglycerides are molecules of glycerol united with 3 molecules of organic acid. When they are forming the triglyceride, they are not protonated, and it's like if they are a base. When pH changes, the organic acid can be liberated and be protonated. Finally, we have nucleic acids that are compounds that have a sugar, which can be a ribose, like in RNA, or a desoxyribose, like in DNA. Sugar gives structure to the nucleic acids. We have the base, which can be cytosine, adenine, guanine, thymidine, like in DNA, or uracil, like in RNA. Bases store the information of nucleic acids. And finally, there is an acidic group of phosphate that can be either ionized or protonated. Normally, this group is ionized, giving the nucleic acid a negative charge. That's why most of proteins that are joined to the nucleic acids, like stones in the nucleus of the cell, are basic proteins. Summing up this part, we can say that pH is minus the logarithm of the concentration of protons. Protons are very important because they are very reactive. And if pH changes, that means that the concentration of protons changes, many body processes can be affected. Principally, we have proteins that can change its structure, its interactions, or its activity. Or you have lipids and nucleic acid that can change its interactions.
If you like this video, please give me a like and subscribe to my channel. In the next video about body acid-base regulation, I will be talking about the origin of pH modification in the body, buffers, how to make a homemade pH indicator, intracellular pH regulation, lung pH regulation, kidney pH regulation, acid-base disorders, special local pH regulation, and the alkaline diet. So I hope you will be following me, and if you have a subject you want me to make a video to explain how it works, please leave it in the comment section. See you!